Okay, so I wanna start with the 2.1 to 2.3 topic questions. If there are any questions, ask. Otherwise, I have a couple that I showed you one the other day, but I have another one that I'd like to look at with you. So did anybody have any questions about any of them? Why can I never find my glasses? Who in here is responsible for my glasses? Can I? Like this is not. I'm not asking much. I'm simply asking my students to keep track of my personal belongings because I'm bad at it. All right. So okay. No questions. I have a couple that I want to go over. So remember we talked about how part of the AP stuff is not just knowing content, but test-taking skills. So let's look at a couple tricky ones. Let's see. There was one, I can't remember which number it was. Yeah, this one. So this says, it shows a cell membrane, phospholipid bilayer, a bunch of other stuff in it. And it says, based on figure 1, which of the following membrane component, no, that's not the one, hang on. No, that's not it, that's not the one I was looking at. Thank you. This one. So it has a cell membrane divided up into 3 areas, and it says, which of the following best describes the numbered areas? So here's our phospholipid heads, tails, heads. And you can look at this and be like, polar, nonpolar, hydrophilic, hydrophobic. You can have a really good idea of how it all plays out and still get this one wrong because it says here, areas 1 and 3 are hydrophilic. They are. C is the correct answer. Areas 1 and 3 are hydrophilic, except C also says, since membrane molecules formed covalent bonds with water. Those aren't covalent bonds with water. Those are hydrogen bonds with water. So C isn't right, even though they gave you half the right answer. So that is so important because I don't do, like outside of my AP classes, I don't do trick questions for my multiple choice because I think it's silly. But they do on the AP exam. So in here, we're gonna have to get used to questions like that. Questions where you are deliberately being led astray and you have to like do the gut check and make sure that you correctly identify that there's a part of this answer that is not true. And then there was one more, I know what I was gonna say. So the other thing is, this first one that I went over the other day, it says, which of the following best describes a cell that is likely to have an extensive Golgi complex, or that's the same as the Golgi apparatus. And what I said is what this question is really trying to address is, what does that cellular structure do? And if you go through the list, you can see that shows up a bunch of times. So mitochondria, what do mitochondria do? What does the endoplasmic reticulum do? This is another Golgi complex. There was a lysosome one. Here's lysosome. So this one is basically, it's not asking the real question that it's asking. What it really wants to know is, do you know what lysosomes do? Lysosomes break stuff down. So you're looking for the answer that ties in with the idea that lysosomes break stuff down. Lysosomes synthesis, nope. Lysosomes storage, nope. Lysosomes protein modification, nope. Lysosomes break things down. You don't even need to understand the rest of the question to recognize what the correct answer is. So those, like, even if you got this one right, which is great, the take home here is start thinking about this stuff, not just in terms of the information and knowing biology, but the test-taking strategery, because it's going to be really important. Yeah? Yeah, number 8. Okay, so this is the, so Phospholipid bilayer and several other components. So which of the following membrane components are most likely nonpolar? And when I did this one, what I did is I know the nonpolar part of the cell membrane is in here. And so I looked for the stuff that was in here. And here's what I came up with. This was my thinking. First of all, cholesterol, because it's fully sandwiched in between the phospholipid bilayer. So I know that that's nonpolar. And then I looked at this channel protein and I thought, okay, well, it might be anchored in through hydrophilic interactions between the protein and the phospholipid heads. It also conceivably could be hydrophobic, and it's kind of locked in there because it's being pushed away by water and there's just no room for these hydrophilic heads to be next to each other. So I thought, well, cholesterol for sure, maybe this channel protein. But I couldn't see the answers. So then I scrolled down and saw that cholesterol only shows up once. And then I was like, oh yeah, well of course the phospholipid tails are nonpolar too. So it's going to be A. Does that make sense? Yeah.
What about the embedded protein?
So the embedded proteins, it's possible, anything is possible. But when I look at this embedded protein, what it looks like is it's locked into place here, which tells me that it's more likely that these are either like hydrophilic interactions or maybe even like some other kind of bond. Now, we know because it's like cell membranes are the fluid mosaic model tells me that it's likely that these are just hydrogen bonds. But you know, if nothing else, it seems to be anchored in place with these hydrophilic heads. Otherwise, it would be pushed by the hydrophilic substances into the middle. It would be in between those two membrane layers. Okay. Other questions? Yeah.
Will you go over number 6?
Yeah. Oh yeah. Okay. So so muscle cells have high ATP demands. And which of the following is a scientific claim about how the structure of mitochondria in muscle cells should be different than it is in other cells because of the high energy demands of mitochondria? So here are the key pieces here. Mitochondria in aerobic cells make ATP, and we'll talk about that next unit, I think, but that's the piece that you need to know for now. Mitochondria make ATP. Muscle cells require lots of ATP. So what we want is an answer that indicates that the mitochondria of muscle cells can make more ATP than the mitochondria of other cells. So let's see what we've got. The inner membrane of the mitochondria should have more folds to increase the surface area, allowing more ATP to be synthesized. That makes sense. I don't know if that's right, but could be. The inner membrane of the mitochondria should be more permeable to large enzymes, allowing the same reactions to occur in both compartments of the mitochondria. I'm not sure that makes a ton of sense to me. I'm not sure why permeability to large enzymes would have anything to do with it. The outer membrane should be thicker, okay, allowing more rapid diffusion. That doesn't make sense. That wouldn't cause greater diffusion. That would cause less diffusion. And then the outer membrane of the mitochondria of muscle cells should have more folds, okay, increasing the surface area, okay, for faster diffusion of molecules from the cytoplasm. So, both D and A make a lot of sense. Without knowing the mechanics of mitochondria, you kind of have an even shot at both of these. In this case, A is the better answer for reasons that you'll learn in Unit 3, because it's the inner membrane of the mitochondria over which that ATP is synthesized. So you wouldn't have been able to know that one. And that's AP classroom for you. So this question was flagged as one for sections 2.1 to 2.3, but it's really relying on information that we haven't covered. Yeah. But hopefully you were able to narrow it down to one of these two, because that's the key piece. That's the stuff we do know. Surface area, diffusion, that we know that there's a correlation between those two things. Okay, other questions? All right. Okay, let's have a little chat. So remember that what I told you the other day is that I'm going to go through this stuff really quickly because you should already know it. And so the goal here is to address questions if there are any, and just to make sure that we're all on the same page. So I'm going to go through this quickly. You're not going to have time to write anything down, and you probably won't need to. But if there's something that you don't understand, stop me and ask the question. So this is the stuff that you all did last week and on Monday in class. Just as a quick review, I'm going to go through this incredibly quickly because this is like even earlier than that. But we have our cell membrane that's composed of these amphipathic phospholipids. This word amphipathic means that amphi means both, and what it's talking about is it has both a hydrophilic and a hydrophobic side to it. I don't think this is a term you'll necessarily need to know, but it can't hurt. Cell membranes are one type of plasma membrane. They consist of that phospholipid bilayer. They are semi-permeable, which we'll talk more about in just a second, and they are fluid, which we'll also talk about in just a second. In the cell membrane, there are other things besides phospholipids, so there's all sorts of embedded proteins, there's embedded lipids, they can have carbohydrates attached to them, so you get those like glycolipids and glycoproteins, where you have something in the cell membrane with a carbohydrate attached to the something that's in the cell membrane. They serve a bunch of different purposes, including transport, which that's what we're looking at mostly right now, communication and signaling, which we'll deal with down the road. There are enzymes that are embedded. Remember, I talked about the idea that they can have these sort of functional chains. So you can have these functional chains. They're not used for transport. They're not used for communicational signaling, but they're used as part of a chain of enzymes that do something. That's how ATP is made. And then they're also used for attachment, which can be cell to cell, or it can be one cell to something else. Our cells are attached to our bones, for example. Okay, and then the fluid mosaic model is the idea that the plasma membrane itself, nothing here is really fixed in place to itself. Things can be fixed in place to this internal cytoskeleton of a cell or to external structures. So it's not that everything is just flowing around willy-nilly, but theoretically it could, and there are some things that do. The membrane itself is held together by what we call hydrophobic interactions. It's not a force that holds them, it's not a pulling force that holds them together. Is that these hydrophobic parts are getting pushed away by all the hydrophilic parts that are pulling toward each other. So they end up getting shoved together because nobody else wants them. All those things that are in the plasma membrane, including the phospholipids, can move around. They can flow around. And then cholesterol, which you see in this diagram as these little structures, that mediates what we call fluidity, the degree to which things flow. It makes the cell membrane more of a liquid and less of a solid, so to speak. And okay, so I'm going to stop there for a sec. That was super review, and now we're going to get to just review. Does anybody have any questions about all of that stuff? Okay, when we talk about cell transport, it's really important to understand the idea of a gradient. And a gradient can happen in a few different ways, in and out of cells. Like this can happen in living organisms and in non-living organisms. A gradient refers to the idea that you can have things that are higher in concentration in some places than in others. It can be a gradual change. So for example, if I had a thing of perfume up here and I did a little perfume, then what would happen is people up here would smell it first, and as it drifts back, you all would start to smell it, and it would eventually reach the back of the classroom. But as it did so, it would probably remain most concentrated up here, at least for a while. So if you were to walk from the back of the classroom to the front, 2 or 3 minutes after I'd spritzed, what you would say is, I can start to smell it, oh yeah, it's getting stronger, it's getting stronger, it's getting stronger. You can also have concentrations across a barrier. And in that case, what you might have is a region where everything is uniformly distributed and it's relatively high in concentration, and a region where everything is uniformly distributed but it's relatively low in concentration. So that would be like, we spray the perfume in here, wait 10 minutes, and it spreads throughout the whole room, and then you go into the hallway. And if you're in the hallway, you might smell it a little bit, but the fact that the air can only move in and out through the door means that not much of that perfume made it out into the hallway. So there's going to be an area in here where there's lots, and an area out there where there's not. That's what generally happens across cell membranes or across plasma membranes in general, is that you'll have a region where there's more of something and a region where there's less. You can also have those gradual gradients. Those tend to happen more inside cells, and they're especially important for biological development. The gradient, the electrochemical gradient in human egg cells and in other animal egg cells is what determines virtually everything about the cells that are in our bodies as adults. That egg cell is one cell. It divides into other nominally identical cells, which should divide into more identical cells. We shouldn't have different parts and pieces to us. We should just be a big blob of identical cells because they all have the same DNA. What makes us different is when those first cell divisions happen, there are different amounts of certain chemicals because there are regions in the egg that have more of something and less of something. And it's that stuff that makes some cells turn into bones, and some cells turn into skin, and some cells turn into pancreases. And those are the only things we have. Humans are made of those 3 things that I said, some of which I already forgot. Skin was one, pancreases was the other. What's the third one? Bones. That's all we are. Bones, skin, and pancreases. Don't you stare at me with your big pancreases. Oh, sorry. I don't know anatomy and physiology very well, just for the record, but I do know that we're more than skin, bones, and pancreases. Okay, so that concentration gradient, if there is some sort of a barrier, then the terminology we use is that we say that the gradient occurs across or over the barrier, that was always confusing to me because that preposition didn't quite make sense, but that's what we're referring to when we say, oh, there's a concentration across this barrier. What we mean is there's more of something on one side, less of it on the other side. And then when we talk about movement of things relative to those concentration gradients, it turns out that it can move both ways. You could have a particle move from where there's a lot of it to where there's not a lot of it, or it can move from where there's not very much of it to where there is a lot of it. But that has to happen in a couple different ways. And that's where the cell transport mechanisms comes in. So cell transport mechanisms are the things that move stuff around in a cell or from out of a cell to into the cell. And this generally what we're interested in is how things move across plasma membranes. That can be the cell membrane itself, or that can be a internal membrane around an organelle like the endoplasmic reticulum or mitochondria. If they move along or with the concentration gradient from higher to lower concentrations, then we say that that's passive. It doesn't require any energy input from the cell. It'll just happen on its own. If we want to move something from a place where it's less concentrated to where it's more concentrated, then we need some sort of like energy to make that happen. Cell transport can happen entirely on its own. The things that move across our cell membranes on their own are things like real small uncharged particles. So oxygen, that there isn't a thing that helps oxygen move across cell membranes. It moves on its own. Carbon dioxide, same deal. When we build up carbon dioxide in our cells, it just moves right through the cell membrane and out. When we need oxygen in our cells, if it's more abundant outside of the cell, it just moves in on its own. And this is something, I'll come back to this many, many times this year, but this is the thing that blows me away most about cell and molecular biology, is that there is no like intentionality to it. It's a bunch of chemical reactions. It's a wind-up toy, so to speak. And the reason I'm mentioning this now is because we're gonna start speaking, you may be able to avoid it, but I can't. We're gonna start speaking in terms of what the cell wants, or what the cell needs, or like, we're gonna talk about these structures as if they're thoughtfully doing things, but they're not. They don't have brains. Brains are made up of billions of cells. This is just one cell. It can't think. It doesn't know what it wants. Things are just happening. And we'll come back to that a lot, because it's one of the most powerful and difficult concepts in biology. When things move passively, we call it diffusion, and this happens on its own. This is a law of physics that is related to thermodynamics and entropy, and basically, if we have something, a lot of something, and it will move to areas where there's less of it. And so there are things that can move directly through the plasma membrane, and those are, in general, small things that are not polar. So oxygen and carbon dioxide are two really good examples. Water is polar, but it's small. And so as it turns out, water can actually get through a plasma membrane. It can move in and out of plasma membranes, but it doesn't do it real quickly. And so typically what we have are proteins called aquaporins that open up little channels for the diffusion of water to occur. And then anything that's much larger than water or is highly polar, so like sodium ions are tiny, but they're charged. They can't get through the membrane. Charged particles, large particles cannot move through the membrane on their own, whether it's diffusion or not, and so they need something to get them through. The something that gets them through are proteins embedded in the cell membrane that lead to what we call facilitated diffusion. So this is for things that can't get through on their, or they can't get through on their own, and that includes things like large molecules, polar molecules. And water, as I mentioned, is a special case because it's small and it's polar, but it's neutral overall. So it can make it through a little bit. If you had a cell membrane with no aquaporins, water would move around to where it's less concentrated, but it doesn't happen quick. And then facilitated diffusion is something that opens up a little channel in the cell membrane to allow some of those things to move around with the concentration gradient. So this would be things like transport proteins. These are generally unique. So they don't let everything in. It's not like a hole in the membrane. It's a protein that will move certain things or allow certain things to move, but will not allow other things to move. And that includes aquaporin, which is for water. And you can, like when we draw these, let me go back here. When we draw these things like this, it makes it look like, well, why can't anything go through there? It's when we recognize that these are proteins embedded in the cell membrane that we realize, oh, here's why. Because it's not just a tube, it's a protein. And what allows things to move through is the shape of that protein and the nature of the interactions. So basically, for reasons that I couldn't possibly explain, sodium ions can't make it through aquaporin. They don't fit, or they stick to it in the wrong spots, or it repels them in certain areas, or I don't know why, but only water can make it through aquaporin. So that's how it works. For things that are needing to be moved from areas of low to high concentration, you can't do that with diffusion. It won't happen on its own. So instead, you need active transport. This moves substances in the opposite way. It takes them from where they are less abundant and pushes them where they're more abundant. And that term push should imply to you that some sort of energy is required, and that's true. And usually the energy is ATP or ATP related. And there's a few different kinds. One is what we call pumps. And so these are things where you have like stuff goes in one way and stuff goes out the other way. In this case, this is a sodium-potassium pump, and it's powered by ATP. We can also have endo and exocytosis. This is where the whole cell membrane is involved in pulling things in or taking things out. There's phagocytosis, which is like large particles that come into contact with the cell membrane. The cell membrane engulfs them and brings them inside in a vacuole. There is pinocytosis, which is similar, but generally involves just like wrapping around a bunch of extracellular fluid and whatever is in there. It can be what we call receptor mediated, which means it can be triggered by certain things, but it's not super selective. Pinocytosis might not happen unless there's something that the cell needs, but it also will pull in some other stuff too. So if it like wraps itself around some potassium ions or something to pull them into the cell, it'll also get whatever else is in the vicinity. Whereas phagocytosis tends to be real specific to some particular large molecule, and it's a little bit more exclusive. And then there's what we call secondary transport. Oops, this is, sorry, this is, my pictures are out of order. This is showing pinocytosis and receptor-mediated endocytosis. And then there's what we call secondary transport. And secondary transport is basically what happens if you use pumps to establish a concentration gradient, and then you use that concentration gradient to move something else in along with whatever is essentially diffusing. So typically the way secondary transport works is that you'll have ATP used along with a pump to establish a concentration gradient. So in this case, there's a concentration gradient being established by this pump where there's more sodium in this area. This is the extracellular area, this is the intracellular area. And then, once there's enough sodium out here, this particular protein will work without ATP. It's using the sodium. The sodium is kind of flowing into the cell, and it's using that diffusion of sodium to pull in glucose. And the way I think about this is it's kind of like if you used a pump to push water up into a bucket, and then there was a tube coming out of the bucket, and you used the water coming out of the bucket to like clean something. So where's the energy coming from? Well, first you establish an elevation gradient. You pump the water up with the pump. That's what's going on here. Once you've got that, then the water comes out at pressure on its own. You don't need to add any energy. You don't need something to squish the water down. It's already coming out at pressure.
So is this mostly used to move certain elements or certain substances to a different part of the cell?
Yeah, so it can be a different part of the cell, but in this case, we're looking at across a cell membrane. So this is in, this is out. So in this case, what's going on is the sodium-potassium pump is moving potassium in and sodium out, and then that concentration gradient of sodium is powering glucose to move inside the cell as well. So the sodium is flowing back in, and that energy is being used to push glucose into the cell.
Does the first pump also use ATP as well? Yeah.
This one uses ATP, yeah.
Does the second one use ATP as well?
No, this one is not using ATP. This one is using the flow of sodium as its power. That's why that's what makes it secondary transport. Primary transport is when ATP or another similar energy containing molecule is used. Secondary transport is when ATP was used to establish a concentration gradient, and then that concentration gradient is used to do this. And some of you might be legitimately asking, well, why doesn't this just pull glucose in? Like, why pump sodium out and then use that sodium to bring glucose in? Just use the ATP and the energy to pull glucose in, idiot. Well, try explaining that to a cell. You can't, because the cell, it can't think and it's not doing any of this consciously. This is the way it evolved. Maybe it's not perfect, but that's how it evolved. Nobody was like, oh, I have a better way. It doesn't happen like that. which again, like, that's one of the things that I think is so cool about all this. Okay, active transport pumps. This is just a little bit more detail about those pumps, and basically what they do is they push things from places where they are lower in concentration to higher in concentration. They generally require ATP. This sodium-potassium pump is a really good example of it because, well, this is sodium-potassium pump is a really good example of it because it's an incredibly important pump for a variety of things, including glucose. But there's also, the last thing that I wanted to mention here, and some of you saw this, is that when we set up these gradients, they're not necessarily just chemical. They can be electrochemical, and there's some physics involved here that you don't need to know. But basically, if you want to pack a whole bunch of positive charges in the same place, you have the same problem as if you're trying to pack a whole bunch of like particles in the same place. They will tend to diffuse away. Positively charged particles repel each other. So if we were all positively charged right now, some of us would be pushed away from each other and eventually like get pushed out the door. That would be a electrical gradient, a charge gradient, where there's too much positive charge in here and things are getting pushed out. Once it's equalized, then everything stays the same. What that means is that those concentration gradients can be either or both, and they might kind of work against each other. Like you can have a real high positive charge outside the cell with a real low concentration of one particular positively charged ion, which means that it would be the balance of which is more powerful, right? Like you're gonna have an electrochemical gradient that would tend to push that particle this way, but a chemical gradient that would tend to push that particle this way. And what ends up happening depends on the relative strength of those two things. And you're not gonna have to calculate that or anything, but you will have to recognize that it's either or both. As you're moving particles around, you might be moving charges around too. And both the particles and the charges contribute to their own separate interacting concentration gradients. Okay, that's my deal. How does that all sit with you? Does anybody have any questions? How many of you, first of all, raise your hand if you feel, you don't have to contribute anything, but maybe I'll check in with you. Raise your hand if you feel like there was something you learned about that was really important that I didn't mention. Raise your hand high. What do you think? Yeah, do you want to, are you okay?
Yeah. I was gonna say like the clathrin protein.
The uniport and symport and antiport, is that what you mean?
I didn't
Oh no, the clathrin, yeah. So that was, yes, and so I did want you to look at that, but that is not something that is specific to the AP class. So what you should have seen was that some of the proteins involved in endocytosis are called clathrin, yeah, and that it's just an example of the kind of protein that can be involved, yeah. Other questions or observations? Okay. And then, was there anything that I brought up that you missed in yours? Yeah, what did I talk about that wasn't?
I'm just like confused why water can go through.
Why it can or can't?
Why it can't.
So basically the deal with water is that it has this slight polarity to it. It's a little bit positively charged on one end and a little bit negatively charged on another end. So typically what will happen is when it bumps up against these hydrophilic phospholipid heads, it'll kind of stick to them and it won't go any further. But those attractions aren't so strong that it can't sort of break through. So every once in a while, you'll get a little water molecule that kind of breaks through, and then it can make its way through this hydrophobic region, and then maybe it breaks through the other side too. That will tend to happen more quickly if there's a lot of water on one side of a plasma membrane. And so water can make its way through the plasma membrane, but because it's sticking to these phospholipid heads, it doesn't move very quickly. So cells that require a lot of water movement across the membrane have a lot of these aquaporins. That just basically creates a pathway, and the water doesn't stick to those, so it can move much more quickly into or out of the cell. All right, so next step is this was something that was originally on the schedule for Monday, but our Canvas outage prevented that. So what I'm going to ask is, first, that you enter the data for your potato osmosis, and that is I made a Google form for you, and the key here is you should all only have two. Some of you, you should only have 3 entries per group. Some of you maybe only had 2, because if you only had 2 group members, you only got 2 pieces of data, but don't do multiple entries of the same one, so you're not all entering it. Just divide and conquer, basically. If all of you do 1, it should work, and all we need is who's in your group, your class period, which solution you personally are entering, and the before and after mass of your potatoes combined. And then that shouldn't take long, but then I have to do some stuff to get that data accessible to you. So while I compile the data, I'm going to ask you all to figure out whatever you can do back there to clean up whatever you can figure out. There is, with the exception of those Petri dishes on the side that have soil in them, I don't think there's anything in there, anything around that needs to be kept. So to whatever degree you can clean, that'd be great. While you clean, I'm going to go through the spreadsheet and put together some class data, and then we'll have the rest of the class to wrap up the osmosis and potatoes activity, which I'll explain once I get the class data sorted out. How does that sound? Pretty good? So good? Have you ever been happier in your life?
Fantabulous.
I'm looking out there, and what I'm seeing are the happiest students I've ever seen in my life. I've never seen a group of students that seem happier. It's so much that I might give you all lunch detention and have you all stay here during lunch today, just because I know how happy it would make you. Okay, if you need to take a break, now's a good time. Let me know.